Nuestro seminario de Frontiers del día de hoy. Y bueno, agradezco su presencia a los que estamos acá y a los que están en Zoom y YouTube. Realmente es un gran placer presentarles al doctor Micha Drukker. Él obtuvo su licenciatura en biología en la Universidad Hebrea en Jerusalén, en Israel, y su doctorado directamente ya en el estudio de las células totipotenciales, también en la Universidad Hebrea. Después realizó una estancia postdoctoral en la Universidad de Stanford, en la Escuela de Medicina, en el laboratorio del doctor Arvind Weissman. Y bueno, pues la investigación principal del doctor Drukker es el estudio de las células totipotenciales y sus aplicaciones clínicas y otras aplicaciones que vamos a ver el día de hoy. Después de su postdoctorado, se incorporó al Helmut Center en Múnich como investigador independiente. En el 2018 se convirtió en profesor y en el 20 se movió a la Universidad de Leiden en Netherlands, en los Países Bajos, para trabajar en el Centro para el Estudio de Drogas y en la Escuela de Medicina de la Universidad de Leiden. Como ya mencioné, una de las principales líneas de investigación es tratar de entender y descubrir los mecanismos que regulan la formación de embriones a partir de células totipotenciales inducidas para crear modelos para estudiar enfermedades humanas y, como verán, también para rescatar especies en peligro de extinción. El doctor y su grupo pues han hecho muchas contribuciones importantes a esta área y quiero mencionar que a lo largo de su carrera como investigador independiente ha dirigido a 18 postdoctorados, 18 estudiantes de doctorado, 9 postdoctorados y realmente ha contribuido a la formación de más gente en esta área de investigación porque da un curso básico de precisamente células totipotenciales en la Universidad de Leiden. Dentro de los descubrimientos más notables, solo quiero mencionar algunos. Descubrió las células totipotenciales que regulan la formación del tubo neuronal y los ejes en el desarrollo. También descubrió las bases moleculares de un síndrome que afecta a humanos, que se llama el Boarding Upside Syndrome. Ha dirigido su participación en la industria es promoviendo el uso de estas células a nivel clínico en condiciones de buenas prácticas de laboratorio para la aplicación en enfermedades humanas y de hecho tiene un término muy interesante que es el uso de estas células como drogas para tratar enfermedades. De los múltiples reconocimientos que ha recibido, solo quiero mencionar uno que me parece muy relevante, que es miembro del Consejo de Investigación Europea. Y su participación en la industria es, ha participado en diferentes comités directivos de farmacéuticas enfocadas a tratar el dolor, inmunoterapia, y es cofundador de Mida Biotech en Ámsterdam y Celidon Therapeutics también en los Países Bajos. Entonces, como ven, su trabajo no solamente tiene implicaciones relevantes en la investigación básica, sino después cómo se aplica ese conocimiento a tratar enfermedades a través de la colaboración con la industria y, bueno, algo más novedoso todavía en tratar de rescatar especies en peligro de extinción. Micha, thank you very much for being here.
Fine, thank you. Don't forget to take the picture. Well, can you hear me now? Okay. I don't think I ever give a lecture when people are 180 degrees around me. It's really special. Take some images, you know, very excited. Okay, thank you for the kind introduction. Thank you for the kind invitation. I will do my best to tell you a little bit today about an idea, a concept that I believe in, and I'm going to try to convince you also to believe in that concept. And maybe it will lead for something greater for humanity, and more, not just humanity as people, as maybe the global system where we live, also for animals. And yeah, I mean, you tell me how it goes, and if anyone is interested also in student fellowships and visiting our labs, you can also discuss with me. So let's start by the topic. I will speak today about stem cells as agents for species regeneration. So basically the idea, and I hope that I captured the idea in this illustration, is basically to put animals that are endangered into freezing vials, right? Most of you are experimentalists, and you go to the lab and freeze cells, typically cancer cells, tumor cells, human cells, mouse cells. But what I'm proposing here is basically an idea of preserving cells for new applications in biodiversity. Now, I will start, of course, with some introduction about stem cells to make sure that we are all on the same page. And I'm using illustrations that are very general, that I use also for very general talks, in order to give you, by metaphors, what stem cells are and what they can do. So perhaps you heard about cells called embryonic stem cells, ES cells. These are cells that we produce, people produce, from cells called the inner cell mass, from a mass of pluripotent cells in embryos. And we scientists, we find ways to fool those cells to think that they receive signals to basically make more of themselves without differentiating, meaning it's something that we call self-renewal. In our jargon is self-renewal. So we can convince these pluripotent cells in the embryos to become embryonic stem cells, and we can make them in any amount. Now, obviously, this is really cool because you can then, if you know development and you know some factors, like shown here, these differentiation factors, you can direct the differentiation, or you at least can attempt and improve the methods to cells that are of interest for you. For example, cells of the central nervous system, cells of the heart, cardiomyocytes. I should have used small short clips that we have that we turn stem cells into cardiomyocytes. They're beating in the culture, or kidney cells and whatnot. So basically, they have the potential to make all cell types in the body. And we as a community can turn pluripotent stem cells quite effectively to different cell types. There's also the other modality, which to us is, and I think it's very transformational, actually, to numerous fields, which and this invention is basically called induced pluripotent stem cells. We take a handful of factors, we do this magic, we call this reprogramming, and we can turn those somatic cells like skin cells, sometimes even urine cells. You can pluck some hair, cells from the blood. You can use many different types of cells in order to reprogram them, and they make induced pluripotent stem cells, which are very similar to, for the sake of the discussion today, to embryonic stem cells. And then, of course, we can differentiate them as well. So that's really the fundamental idea of the pluripotent stem cell field. Now, what I'm portraying in this slide is something that I think is very conceptual to understand that this pluripotency that we capture is something that is very transient in embryos. So this function here that I tried to illustrate, maybe it's not the best illustration, but I did my best, is very transient in the lifetime of every individual. So pluripotency exists for approximately 3 days in your own natural history. If this is your life, it existed when you were that small for approximately 3 days. And then it perished. We don't have pluripotent cells anymore in us. And the reprogramming can take, basically is sometime along our lifeline, we can reprogram cells into pluripotent stem cells and they become self-renew, as I said before. And I always try to basically find a nice analogy for this. I always think about this as Back to the Future. And I hope that most of you watched the movie. If not, watch the movie, basically because stem cells what they do is basically they stop time, they renew indefinitely, and we can then tweak them to become things that are useful for us. This process is, I'd say, I haven't checked with all mammals, but it's very universal among mammals. The pluripotent in transit in an embryo. This is an illustration of a generic embryo, but under the assumption today, let's say that every species will, of mammal will have a very similar say mechanism of self-renewal that we can reprogram during its lifetime. This is the typical topic that I talk about. My lab works in an area that I call cell pharmacy, and that's a discussion that we had yesterday here. I see some nodding, which is very nice, so students remember. Our vision is to turn stem cells in the future to a pharmacy where it's of course a metaphorical pharmacy, that we will be able to create spare parts for people, and as well as to study disease and to, for example, test toxicities, many other things by having this cool ability to make all sorts of cell types. And now thinking about this concept for many years, it occurred to me that maybe we can do things that are actually very useful as well, not just for basically our own medicine. And that's our topic for today, how we can use the biotechnology that is behind this concept of cell pharmacy to better the world. And among the many papers that we publish about how we study development using stem cells and what we can do with them, I always had a hobby, and I still, of course, have this hobby or pet project to create stem cells from different species. And I will talk today a little bit about this paper that we published, where we made induced pluripotent stem cells from an endangered species where there are only 2 remaining animals in the world. So after these 2, the species will not exist anymore. It's functionally extinct. 2 females, by the way, they are infertile. So that's the end of the species. So how can we harness? The idea is to discuss how can we harness this idea for the sake of not just people, but actually more ourselves within the general notion of the world with animals. And this is important because it's not just the northern white rhino. We are experiencing an enormous increase in, or I'd say accelerated rate of global extinction, one can say, of species. And I suggest, I mean, for those of you who are interested to read this book, The Sixth Extinction, because we are now at the 6th amplitude of mass extinction in the history of history, not history of mankind, history of history. So this is becoming really bad. We are, unless we act urgently, we will lose many of the species, and actually, I think most of them in the coming century. And the problem is this, that the conventional conservation efforts are unfortunately insufficient. Poaching, yeah, you know all those things, I don't need to tell you, but perhaps you're not aware how severe it is. It's very severe. For example, this is a slide that I prepared while attending a conference in the Rotterdam Zoo last year, and listening to different groups who work on different types of animals within the zoos, trying to understand the problems and maybe help them by stem cell technology somehow. So I just listed a few, I mean, there are many more, but this is just from one afternoon discussing elephants and Asiatic lions and black rhinos, and realizing that although we might think in the public eye zoos are important and conservation efforts are taking place and are successful, it's not so much the case. For example, elephants, they suffer enormously from herpes virus, for example. And so about 1 to 10 or 1 to 5 of the newborn elephants are dying in zoos, for example. Asiatic lions, at least in that zoo, but I think in actually many zoos, they're a very small population. They start showing very early on phenotypes of ataxia. So of course, they then don't breed well. And eventually, we think that these species of lions will go extinct. Black rhinos, this is the most, let's say, most abundant rhino. 30% are affected by weird weird syndrome called iron overload disorder. Makes basically the animals very weak. They don't reproduce well. So although you sometimes see images of success in zoos, it's actually very hard, and it seems that it's fragile, very, very fragile. So I wanted to focus today on an example, and I'll take the rhino as an example. This is a species or genus that we work on for some years, and I already mentioned the northern white rhino, where there are only 2 animals alive today. I said that they are both infertile. There are only 2 of them left, and once those die, that's going to be the end of the species, unless we are successful in doing what I'm going to show today. Southern white rhino, there are about 10,000 of them. Black rhinos, actually, black rhinos are 3,000 or so, more or less. And you can see the populations of the rest. For example, Sumatran rhino, 30, Javan rhino, 80. So we will lose, very likely, a couple of the species of rhinos. And again, this is to give you the idea. It's not necessarily to cover all endangered species because they are numerous. And now, in preparation of this talk, I, of course, Googled a little bit and used Wikipedia and said, okay, why is it, to try to convince you, why is it important to Mexico? And it's very important for you, I think, because you have enormous biodiversity in this country. I mean, about 10, 12% of the species, actually, of mammals and actually animals and plants are represented in Mexico, which is amazing, 10%. And today, I even learned about this amazing mammal, how do you say, vacutir? Thank you, everyone. So now, this was a test to see if you are still awake. Yeah, there are about 10 of them, I think, left. So basically, it will go extinct unless we do something. And we know that conservation efforts, I mean, in many cases, they fail. So what can we do about this? So what I would like to propose today is that you, or basically us, take initiative and think about stem cells as agents for species revival. I call this also species regeneration. So not to regenerate a tissue or the blood or part of the brain, but actually regenerate a whole animal community. And of course, this follows narrative that exists. For example, seed banks that are used in order to, or basically they are collected in order to ensure food safety, for example, in the future. But I think this is easy because seed banks already contain embryos, because seeds are embryos basically. We need to do much more. We would like to collect cells, turn them into embryos, okay, with our biotechnology, which is much more challenging, I think. But basically, the principle is the same, to have this sort of a seed bank for animals in the future. You may have heard about this company, for example, by George Church, called Colossal Biosciences. They are thinking about using DNA in order to bring back the woolly mammoth, for example. I'm somewhat skeptical, let's put it this way. But still, it's an interesting concept. And for that, obviously, we don't have cells of the woolly mammoth. We don't even have nuclei frozen. In Siberia and other regions of the world where mammoths, you can find tissues of mammoths, most of the cells are actually, in fact, they have undergone some freezing and thawing cycles. So basically, there's no intact cells in the permafrost. In contrast to you, because we see tissue and we think the tissue has intact cells, but actually, no. So it's going to be quite an effort, I think, to try to go from ancient DNA for the extinction, for example, woolly mammoth. But whatever the case is, even if George Church is able to synthesize chromosomes, you still need to make cells, right? And you still need to, from the stem cells, you need to make eggs, and you need to make the animal. So probably Colossal Bioscience will basically piggyback on such developments that we also work on. So the idea is that pluripotent stem cells that we said that renew in culture, whether they're induced pluripotent stem cells or embryonic stem cells. I'll show the two options. They can differentiate to all setups on the body, and in the female body, obviously, females also differentiate cells that make eggs. We know that very well. And then these eggs, these oocytes, can be fertilized and basically give rise to you guys. Now, what has been shown, and I will show a little bit more of the data for that, by our colleague Katsuki Hayashi in Osaka University, who we collaborate with. He actually showed that this whole prospect actually works. He can make a mouse from mouse embryonic stem cells or even a skin fibroblast, going all the process and making a new mouse. So this, what I'm telling you, this idea is proven in mice. It's possible. Going from skin cell all the way to stem cells, oocytes, implantation, and giving rise to a live birth of animals. So it's not basically a fantasy, it's reality in mice. So the idea is to basically to create, to power conservation efforts by using synthetic embryos, or at least to enable this. I'm not selling you the idea that you should start growing here mammoths or whatever, you know, dolphins or whatever, but at least what I'm trying to convey the message is that we should keep, for future sake, cells representing as much the broader population as possible in order to, for the future, to enable that idea to go from stem cells to animals. And as always, I always give homework to students to watch movies, and they like it a lot, I hope. So in Avatar, in case, I'm pretty sure that most of you watched Avatar, and you haven't watched Avatar, it's an awesome film. Watch both the two movies, they're both excellent. So in Avatar, there was a scene where you couldn't hear it because of the noise, because of the fans, but Avatar shows that basically portrayed the notion that tigers have been remade, de-extinct, they were de-extinct by using cloning. So that's actually portrayed in the movie. But I want to give a very important message across that our idea is not to clone, okay? Our idea is to basically make oocytes and gametogenesis of sperm, and that will create a population of that is diverse, because obviously we are not like our siblings. And this is shown here. And I'll go for the steps in a minute. I just want to say that I'm not doing this by myself at all. We are working in collaborations with many groups. One of the leading groups of veterinarians that work on this topic is the group of Thomas Hildebrandt in Leibniz Institute in Germany. We work very close to them. And together we came up with an ideas plan to how to save the northern right rhino. And again, it's not just about the northern right rhino. It's basically to create the idea for people to follow, because the problem is much bigger than just the northern right rhino. And this is an illustration that was published in Nature, covering some of our studies, where basically showing this idea. So if you want to orient yourself easily around the topic, you can look at this review. The idea would be to make artificial gametes from stored tissues, as I said, producing induced pluripotent stem cells, and then cells called primordial germ cells. So these are basically the offspring making cells, in case you are not very familiar with developmental biology. Primordial germ cells in the female will differentiate into oocytes, and the male will make sperm. If you are able to do that, combine it with sperm, actually frozen sperm, for example, or frozen eggs, you may take it to the next step to try to transplant an embryo into a surrogate female that is close by. So the idea, of course, is if you are lacking, if the species is so thin, or there's so few animals, is to use surrogates from closely related species. And if everything works very well and you pray very hard, maybe you'll get a northern white rhino. So that's the idea. And that's again the notion that I wanted to clarify here, that we are not cloning. We are basically via meiosis and gametogenesis. We are aiming to create a population that is more diverse and not all clones. Like, for example, Dolly, that you must be familiar with, and shown next to Elon Wilmot, who passed away recently. He was a true pioneer in this field. We can also talk about this genetic variation later. So what's also important to say, and I'm sorry for this long introduction, just want to lay all the arguments, that there are additional motivations for establishing so-called an iPSL zoo. And I'm very fascinated with many of these topics because the classical way that we study, for example, natural history, we go to museums and we compare anatomy and morphology of different animals, but we don't really understand how it comes to be because we cannot study a whale as it develops, or a giraffe, or a lion, or a polar bear, or whatever. So I think this idea is a very powerful way or to power something that I call cellular natural history, or maybe stem cell natural history. I think it would be amazing if we can compare all sorts of aspects, whatever you are interested in. There's also, of course, another topic which is disease research. People get disease that pathogens that come from obviously from animals. We all heard about coronavirus, obviously. So imagine that you have in the fridge stem cells from different species, and then you can quickly test, for example, coronavirus and understand its function, not necessarily by establishing models, but actually having the cells in the fridge. And of course, there's agri-tech and alternatives for animal farming. Maybe you want to make a steak out of a lion. That can be an interesting idea. So that is also a prospect. On this direction, my lab is actually works quite extensively in the cellular natural history, because I'm very puzzled about how do we become to be right, left, back, right, up, down. I'm very tall myself, obviously, as you can see, here in Hawaii. So I'm very curious about, how we are made, the way we are made. And this is something that if you have cells from different species, you put them in culture next to each other. If you polish the system well enough, you can say, okay, what's similar, what's different, what is maybe controlling processes. So we have a couple of studies now. For example, one is in review in Cell on the body axis forming cells. For example, we obtained cells from whales. I'm very, very curious to see why whales are so big. What actually drives the size of whales? Unless you have cells in the fridge that you can obtain and study, it's going to be very hard with the classical way of natural history. I would like to understand the mechanism. So what are the molecules behind that? We have more studies that will come out on longevity. We are very interested in the intrinsic longevity of cells. So how long cells are able to live from different species. Of course, we are not waiting decades for that. We have ways to accelerate aging and to compare species in that sense. I came here for this Frontiers in Genomics program, and I wanted to say that we do a lot of work on genomics as well, and phylogenomics of different processes. But then what's really cool, that we can go to the fridge and take stem cells from mice, from a naked mole rat, from a rhino, from the Queen of England, from whale, and then placing them in culture and asking, okay, can we validate an hypothesis with this approach because they're sitting in the fridge? Otherwise, if I had to go to the Queen of England, she would not let me experiment on her, obviously. I put her there because of longevity. She lived very long. Anyways, so this is one grant that we obtained for that purpose. Okay, let's go a little bit more concrete on this iPSL business. And I want also to give a little bit of education with respect to what this entails. In case someone is interested to pick it up here, it's very complicated. But you have to obtain, for example, permits to capture cells, for example, from this beautiful dolphin that I didn't know about, the amazing dolphin. You need to arrange for animal biopsy collection. That's not so trivial, obviously. If you need a biopsy from a whale or from an elephant, I'm not sure if you have elephants or whales on the campus. You need then to develop ways to, you need also sampling handling, transfer, you need to think about that. To obtain cryopreserved cells, to produce iPS cells. I'll show you some productions of iPS cells from understudied species. And there's no, let's say, good manual to follow because we don't really know what works for what species. But nevertheless, it's possible. And then the idea would be to differentiate to egg and sperm and embryo transfer. And the expertise you need is very broad. You need legal advice. You need to speak to wildlife caretakers like veterinarians. You need a lot of genomics. Good for you that you have all these tools here in bioinformatics. And then you need people like me that know some things about cells. I'm not an expert in genomics. A lot of collaboration and hoping for the best. This is how it looks in the field, more or less, even just to, as a procedure, just to take an egg out from a rhino. Look at the team. So basically, like 30 people have to fly all the way to Kenya. All expert veterinarians from the Thomas Hildebrand group. Yeah, just imagine, looking in an elephant or whale for one egg after superovulation. It's not trivial at all, but it's possible. They accomplish that. So all these artificial reproduction technologies, they are advancing actually fairly well, mostly thanks to Thomas and his team. And if you want to look at this project, this is a really amazing project called the BioRescue Project, where we collaborate. Yeah, so I put this here. So basically, we are together. It's mostly led by Thomas, but we are also helping to create numerous freezers where the iPSs are or the fibroblasts are. For example, there in Germany in the Leibniz Institute. And I show this picture because sometimes I give these lectures in Germany, and this is a very famous polar bear. You will not know because you live in Mexico, but this is a famous polar bear called Knut. Everyone knows Knut in Germany because it's an icon. And we have Knut in one of these freezers. So that's why it's here. A little bit about reprogramming. Reprogramming, and we had discussions today, comes with many flavors. And it's a very detailed topic of how, and that's what we discussed yesterday with students. So what's the depth of reprogramming? What are the methods of reprogramming? For example, using mRNA technology. We can today, like the way that you receive coronavirus vaccinations, in a very similar way. Actually, we did those things even for the rhino before the corona pandemic. Okay, so we reprogrammed rhino cells to iPS cells with the mRNA technology before any of you heard about corona. These kits, or basically these developments of different reprogramming factors, we spoke about KLF4 today, and Micha and others, you can make them by RNA, but you can also make them as plasmids or whatever. So it's very, it's a big topic and many options. Biopsies are a very interesting topic as well. You can, as I said, start from skin, from blood, from urine sometimes. We have colleagues who actually collected, it was unbelievable. I was one of the mentors of the student, and I said, it's never going to work. So what they did, they wanted to make a chimpanzee and orangutan stem cells, iPS cells. So what they did was actually one student went to the zoo and collected urine from the floor. After, the monkey was whatever, what the monkey does, or sorry, the ape, collecting some urine. It's, of course, extremely contaminated. Yeah, we are using those cells. They're amazing iPS cells now. I thought it's never going to work. I think it's published in Stem Cell Reports or Cell Reports. I don't remember, but I can show you this study. How do I go forward? Yes. And that's just, to give you a little sense. For those of you who didn't see how it happens, maybe I move out of the way. This is cellular reprogramming. It's something that, and we spoke about this yesterday, the whole class was about that. It's very difficult for cells to reprogram. It's very rare. You will see only 2 colonies emerging. All the rest of the cells will just die at the end. We spoke about this yesterday. Just imagine that you have an identity of a fibroblast or a blood cell, and you need to basically open up all the chromatin. You can see it's already about 2 weeks, almost 2 weeks of this movie running. Still no stem cells. And now it's coming here. Only 2 cells from all this field became pluripotent stem cells. And this is for a species that we know how to reprogram. This is human fibroblasts. And that's actually newborn fibroblasts, which are much easier than older fibroblasts from old person like me. So you're creating in this process highly confused cells, right? They're highly confused. They think they're fibroblasts, and you put all those things there to try to access the genome and metabolize it. That's why it's very inefficient. But the bottom line, that it's actually working. To expand the next slide of slides, I want to say very briefly that pluripotent stem cells exist in 2 main flavors. And excuse my jargon now. There is a flavor called naive or ground as well. You can think about them as earlier type of pluripotent stem cells. And there are also more adult or more advanced type of pluripotent stem cells. They are called prime pluripotent stem cells. And intentionally, I placed here these icons. So conventional, in a conventional way, the conventional methods to reprogram and to culture human pluripotent stem cells would be to the prime state. In the mouse, the classical studies always have been using the mouse naive pluripotent stem cells that resemble more the cells of the blastocyst, the earlier stages of the blastocyst. So pluripotency is not just black and white, either you have it or not. It comes in flavors, okay? And those flavors matter because it also tells you, for example, how deep the reprogram is, how successful it is, and whether the cells can differentiate to all cell types of the body or not. Maybe very briefly, and we also studied this yesterday in detail with the students here. We have optimized conditions. Maybe look at the column. This is columns. We have optimized conditions to promote the self-renewal apparatus, the cells in the naive and primed state. Later, differentiation takes place. So we know pathways that basically hack the cells, and convincing them to make pluripotent stem cells of naive or ground state cocktails. So the question is, how many iPS cells have we made? I mean, in general, I mean, the community at large, we're actually working on a review to describe that. Pluripotent stem cells so far have been made only for mammals. Okay, no birds or all the rest of the amphibians, sharks, and nematodes, and so on and so forth. It's possible now only for mammals. It's another topic for discussion, but I think that maybe there's a chance that it will happen in birds. I think there are studies that will show that it's possible with birds in the coming years. Let's keep that. Now, this is also to show you what IPS is now narrowing down or focusing on mammals. So what mammals have been reprogrammed in general and in our lab as well. This is a really cool animal called the elephant shrew. It's very close to the elephant, but it's not an elephant. And it's also not a shrew, okay? It's not a classical shrew, it's an elephant shrew. I don't know who picked up the name. It's also called sengi, this one. It's very close to the elephant, but of course, elephant is like, I don't know, 10,000 times bigger. It's interesting to learn that elephant shrew and tenerax and more older type of mammals, actually, there are no stem cells yet derived from them. So there was a huge clade of mammals where stem cells have not been made yet, but we were able to reprogram the elephant shrew recently, and we are about to publish that. So the first Aphoteria induced pluripotent stem cells. We also have in the lab, we didn't make it ourselves, but collaborators provided us with bat iPS cells, rhino iPS cells that we made, mouse, of course, naked mole rats that we obtained from others, and so on and so forth. Many non-human primates, chimpanzee, gorilla, and so on and so forth, also human. And we obtained also cells from whales and lions and polar bears. But the reprogram will await for students to come and put their minds to try to turn them into iPS cells. Let me show you some data that what we've done in this collaboration about the rhinos. In this collaboration, I told you there are 2 specimens left from the northern rhino. They look very tired already, right? So they will go extinct, and you can see that they are rather unhappy about that. They don't look very happy, Fatu and Yamin. But we actually, in this process, we obtained fibroblasts from an animal that actually passed away recently. Her name was Nabira, and we obtained her fibroblasts and started this journey of making iPSs. Sorry, before that, I should say that as a step towards reprogramming, we also optimized culture conditions in a way, or tested different cocktails in order to reprogram, by reprogramming southern white rhinos, ES cells, okay, not iPS cells. So we were able to obtain, together with our collaborators, you can see, it's very hard to see here, but this is a procedure where they isolate eggs from a rhino, from the southern white rhino, This is shown a bit better. They were developing nicely in culture to blastocysts. And then in this collaboration, we were able to derive embryonic stem cells, okay, which we said are very similar to iPS cells. So this was as a proof of concept in Nature Communications that we published 3 years ago. And then we said, okay, nice embryonic stem cells from the southern right rhino. And they looked to us, and some of the characteristics indicated that they are similar to the human prime state pluripotent stem cells. Again, we didn't know ahead of time. Couldn't really know. We just tested things. So the question was, can we make naive pluripotent stem cells? And naive pluripotent stem cells actually matter because they tend to differentiate better to primordial germ cells. So we said, okay, let's try to make now northern right rhino and turn them into naive cells. And that's what we published here in this study. This is Nabila that unfortunately died. She was the third last northern right rhino. We programmed her cells, her fibroblasts, using, this is all published, using episomal plasmids, so not mRNAs. We already also did it with mRNAs later, but in this study with episomal plasmids, we showed that they are pluripotent by the standards that we apply. It doesn't really matter to go into the details, but there are genes that are, and proteins that characterize the pluripotent state. But as I taught the students here yesterday, how do you prove pluripotency? Anyone wants to say? The students know. How do you prove the cells are pluripotent? We said not by staining markers and saying some markers. Anyone wants to say? Said it yesterday. We learned it yesterday. We said should always, you remember the axioms that we yeah, I'm looking at you. Remember the axioms that we discussed? The third axiom was stem cells are always retrospectively defined. After you were also in the class, right? Gustavo, you should know this. But how do you test that actually in the rhino? How do you prove that a rhino is a pluripotent stem cell indeed? Yeah, it's not easy. You can make teratomas as we discussed yesterday, but and perhaps make embryonic chimeras, which is also very difficult. I'll talk about this a little bit later. But maybe there's another cool thing that one can try, maybe to create trans-species chimeras. It wasn't perfect at all, okay, but it worked to some extent. Because what we did, we made these naive-like cells. We transformed the pluripotent stem cells that are human-like to the naive-like with some tricks that we have in culture. And then we asked, these are rhino iPS cells, I'll remind you, rhino naive iPS cells. We transplanted them into mouse embryos, okay? We made chimera, human rhino chimera. Of course, we didn't make new bones from that. We just let them grow for, I think, up to E7.5 or so. The data is not shown here, but we were able to show that the cells are more potent in their differentiation potential by contributing much more broadly to the embryo if the cells were at the naive state compared to prime state. So we showed that basically the potency of the cells mattered for the competence to differentiate, and we've proven that by this case experiment of injecting cells from a rhino into a mouse and growing it into the mouse in the mouse uterus. When I suggested this experiment, they told me that I'm completely nuts. They were right, I'm completely nuts, but it worked. Through these collaborations and work, we now established the northern right rhino, iPS cells, southern right rhino, embryonic stem cells, I think also iPS cells by now. We, together with our colleagues, we published recently the Sumatran rhino as well. It's also highly endangered. It was published in iScience recently. And the other species, yeah, I mean, we will come to that eventually. So is it feasible to then make gametes from iPS cells? I already spoiled it, and I told you, yes, it's possible in the mouse. And I'll very briefly mention how. This is work by my colleague, Katsuya Kobayashi from Osaka University. If you're interested, this is a really amazing piece of work. Katsuya is really a magician of primordial germ cell differentiation in vitro, okay? So he's able to turn blastocyst-like cells, or inner cell mass, or pluripotent stem cells, to primordial germ cells, and then into eggs. I'll go through this quickly because we are already late. I'll show that that he is able to aggregate primordial germ-like cells that were made completely in vitro with gonadal cells, and they developed to things that look like eggs. But not just that, they can be then fertilized with sperm. So meiosis one cell, then 2 cells, so they are developing in vitro, making blastocysts, so embryos. And I remind you, it started from mouse embryonic stem cells or from iPS cells, and you can obtain live births. So you can get animals, and these animals are fertile themselves. We also spoke about this in the class yesterday, Gustavo, right? We said you have to make all the tissues in the mice, and they make these skin-derived or embryonic stem cell-derived made into eggs in vitro. They can make offspring. Very impressive, I think, extremely impressive. Now, together with Katsuya now, and this was just was recently published, we are working together, of course, on the differentiation of the southern and the northern right triangle stem cells into germ cells. So primordial germ cells. We're trying to turn those cells into oocytes of rhinos in vitro. And this principal component analysis, which I don't have time to explain, shows you this differentiation pathways are in the same direction, indicating that when you make primordial germ cells of the mouse, that I just showed you that are functional, and very similar human ones, the rhino follows similar transcriptional trajectories, indicating that we are in the right direction to make primordial germ cells, also from the rhino. This is a very recent development, embryo transfer technology, and I wrote this actually while I was on the way here. Over Christmas, I was in touch with our colleagues a lot because southern white rhino successfully where an embryo transfer has been accomplished in the southern white rhino recently. However, the female unfortunately died a few weeks later. Her death has nothing to do with the procedure. It was some bacterial infection, or viral infection, I'm not sure, some pathogen. It has nothing to do with the procedure, but she passed away, and but that actually gave an opportunity to open her up and to see an embryo that was made by embryo transfer in the rhino. Okay, so we can, if you have a candidate embryo that you made in vitro, we know that they can be transplanted into a pseudo-pregnant female and make a baby rhino. Well, I'm almost out of time. I'll just say that I wanted to mention, I mean, I think this importance and proof of concept of these studies. We haven't made the rhino yet. Hopefully we'll make it eventually. I'm pretty sure we discussed this yesterday. I was betting between 5 to 10 years, but let's see if I'm right. We said that the motivations, and actually this can be used for research that is much broader than the species revival. For example, the cellular natural history, and I would like also to mention very briefly disease here, because I mean this is an interest by many. Infectious disease, for example, COVID-19 pandemic, and there will be many more pandemics in the future, we are pretty sure. So very recently, our colleagues were able to, Thomas Waka, made bat iPS cells, and actually, and you know that actually bat cells have been proposed as basically the host of COVID, and his analysis is not mine, of gene ontology analysis and gene ontology analysis actually showed that coronavirus associated pathways were highly enriched in the pluripotent stem cells of the pluripotent stem cells of the corona, sorry, not the corona, of the bat iPS cells. I highly suggest that you take a look at this paper. It's very fascinating, indicating that in this population, the genome is already geared to deal with coronavirus somehow. But this is still, I mean, an open question, but very interesting for this type of research. So, coming to the end, I hope that I was able to tell you a little bit about iPS cell technology that's beyond the conventional, let's cure someone with diabetes or Parkinson's and so on and so forth. And I would advocate or basically suggest, you know, that you or next generation here will try to perhaps this effort of live cells and preservation of these live cells, maybe even stem cells here in Mexico, because you are basically, I see it as you were given this wonderful biodiversity, and maybe you should, stewards, take care of it. I would like to thank a few funding agencies like John Templeton Foundation, also the Volkswagen Foundation supporting this type of work that I showed today. These are the main funding that went into this sort of studies. Here I should mention mostly our iPSL co-facility still in my lab in Munich, Iona Rusha and Paul Iksani worked on this, a lot of the results that I showed today. Also Dordenlai Kelly and Alexander Del Porter. And as I said, our collaborators in Berlin, for example, Sebastian Dicke and Thomas Hildebrand contributed, enormously in pushing this project forward. And with that, I would like to thank you for your attention, and I'm happy to take questions.
¿Preguntas?
Well, first of all, thank you for this wonderful talk. And well, I have three little questions. I don't know if they are too much, but well.
I'm here, so go ahead.
All right, perfect. Well, my first question is regarding the endogamy problem, because especially
can you say that again? I have trouble. There's many fans, so please speak up.
Is regarding the endogamy problem, because well, you're starting with a very small genetic pool of only two females, two sterile females, and well, if you were to regress those pluripotent cells into a oocyte, they will have a very small gene pool. Even if you were to utilize the frozen sperm of those males, the gene pool is still very, very small and will create eventually in perhaps not the next generation, but 2-4 generations in the future, a very serious endogamy problem. How would you address that?
Okay, so I'm here. I have a few answers to you, and actually, I prepared the slide because I knew it's going to be the first question. So I knew that you're going to be here and ask this question. So, but I'll answer it in few dimensions. Okay. Right. First of all, this is technology development. I'm not just saying that. It's going to be the northern white rhino. This is basically to the world to raise awareness. As well. So rhinos are, by the way, are lovely animals. If you get to pet one, very soft to touch, very interesting animals. But there are other species where there are more specimens you can collect now. So that's the first answer. Second, actually, you'll be surprised that the genetic variation or the diversity among northern white rhinos, we have tissues for 10 of them or so, is actually greater than the genetic diversity in the southern white rhino. So they are representing a very large population from different regions. Don't ask me why. This is really beyond me, but people have sequenced their full genomes and their claims as such by very serious people. So even there are few, maybe they represent large genetic diversity. Okay, that's the second answer. The third is that iPSCs actually, you can start with those, infertile things, right? So these individuals, and actually increase the gene pool. So say you have a lions, 20 lions, you have another 20 lions that are infertile, but you can bring them back to fertility because they are not fertile, because they are not in the mood or whatever, right? So basically, if anything, iPSC potentially will increase the genetic diversity compared to, say, in a way, a breeding program currently, right? And breeding program is with the endangered species very small. With the Simpsons, maybe you even expand over it because you can take, a dead animal, you can take an animal that doesn't want to breed for whatever reason, that doesn't like its partner. So that's my answer to you already.
Right, thank you very much. My next question is, well, you're saying that you're going to implant the embryo of a northern white rhino into the uterus of a southern white rhino mother. But as far as as I know, I could be wrong, there's a distinct genetic differentiation between the two species. If said differences is great enough, it could create a reject of the embryo of the surrogate mother. How would you address that?
So you're right. There are, of course, differences, but actually, we are all very different from our mothers, right? I mean, enormously different from our mothers without being a cross-species. And actually, embryonic development takes care of that, and actually the mother takes care of that. There's a tissue called the sidua in the maternal interface. And I'd say that I'm not so worried about that, actually, or not worried about this at all, because embryos are tolerated very well, and whether it's the same species, and I think it will be the same case of cross-species, because inherently you have hundreds of antigens, and MHC molecules that are very different from your mother. If you take some tissue from you and whatever, I mean, transfer it to a relative, it will be rejected immediately. So the embryo takes care of that, actually. The placental development takes care of that. It's an issue to remember, but I don't think it's going to be a major issue.
Right, thank you very much. My last question. You said that this process is going to be restricted mostly to mammals, perhaps birds and reptiles in the future. But, well, there are a group of animals called Cnidaria. They are already able to do this process in a more natural way. I don't know.
How are they called?
Cnidaria, jellyfish.
Jellyfish.
Yeah,
okay.
jellyfish. I don't know if you've heard of this species, Turritopsis dorini, or commonly known as the immortal jellyfish.
Yeah.
It's a species that is able to regress back into a polyp by de-differentiating. Well, the term is transdifferentiation. It's specialized adult cells into their more younger pluripotent stem cells. Could you take this process perhaps apart and apply it to the other species that this particular process could not apply to?
So this is a very good question. The answer, I don't know, okay? I think, so Hydra and these marine organisms, I mean, they're also a passion of mine. I grow actually corals and anemones at home. This is a big hobby of mine, and I'm very interested to understand how basically these developmental processes actually work in asexual development. And there's not much known about this. But I want to say something else. I mean, with respect, okay, jellyfish is nice, right? But they are very different from us, extremely different from these larger animals. Let me just say one thing that I think is fundamentally important here. In a way, mammalian development works in a stem-like logic, okay? Because embryos are very small, they contain some cells, so there's some self-renewal, some differentiation, some self-renewal, some differentiation. If you look back to reptiles and amphibians and fish, development works in a different way as far as I understand it. You make many blastomeres, and then kind of like a little bit like the insect, we basically make domains, anterior, posterior, and so on and so forth. So there is not much of a stem process there, okay? And the chicken is more or less between, because they're made from dinosaurs, and we also came from basically the same root that dinosaurs emerged from. I think it's possible for birds. I would be, actually my students are convincing me to try now with chameleons. They're trying with chameleons in my lab. I told them I don't think it's going to work, but I'm not going to limit students not to try. I think in order to do this, you really need the logic of self-renewal to be in place. And the Hydra, and that's a de-differentiation. This is something I think very interesting. Maybe you can learn for this, but it's fairly, I don't see exactly how we can apply it here, for example, for fish, but maybe it's possible.
Right, thank you very much.
Las personas que están en Zoom o en YouTube pueden escribir sus preguntas en el chat y aquí las leemos.
Sorry, that was in okay, sorry, I thought that this question is Spanish to me.
I have two questions. One is, as you know, the major difficulty to maintain and generate pluripotent stem cells is that those cells retain the capacity to produce germ cells. And as you know, embryonic stem cells are mostly with the induced pluripotent stem cells. They can produce all cell types, but it's difficult that they retain the capacity to produce germ cells. Then I don't know if it's a way to measure that capacity, because if that is not gained or is lost during the production of the IPS, then when you will have a cell capable of producing many cell types, but you will not reproduce the animal itself.
Exactly. So let me answer this briefly. So that's exactly the motivation to make naive type of stem cells in the rhino. because the general knowledge, and we haven't proven it yet, but the general knowledge is that they will have a broader competence that includes, as you know, you're a mouse developmental specialist, but still it's just a mouse. I mean, the other animals accomplish their development in different ways. Our basically thought process or working hypothesis is that the cells if they are more naive, more like the mouse, that's what we created in the rhino. They have a broader differentiation capacity, including germ cells. And we showed that they have higher tendency to create more tissues in the mouse chimera. We showed this and we published this already. So you're right. I mean, we'll have to account for that. But it's not that we are going to accomplish everything in just one go.
Okay. And in the same sense, I believe that you have to think that if you are going to derive IPS, you have to use a male, not a female, right? Because from the male, you can produce the two gametes, but from the female, you will not be able to produce a male, right?
Yeah, but how would you produce a female from male IPSs?
No, because if you start with a male, you risk there are papers that you can, from a male, you can produce a female.
Yeah, I mean,
but if from a female, you cannot produce a male because you will not have a white
not yet.
chromosome.
I mean, maybe, yeah. I mean, yes.
There is no white chromosome.
Yeah, I agree.
So
But this is, I mean, that's a good question. But I mean, we don't take it to that degree of complication. I mean, we start with male individuals, female individuals. We try to conserve all of them. I mean, in these cryotanks. And I mean, there are different differentiation processes for sperm and for oocytes. So I'd say in an ideal world, we'll be able to basically to have males and females separate and differentiate both. But actually, sperm is easy, as you know. So even for the northern white rhino, there are numerous frozen sperm specimens from the northern white rhino. So I worry less about the sperm, let's put it this way. I don't worry so much about whales.
Okay, hi. This way.
Oh, okay, sorry. I was looking at
Thank you for your talk. I think it was really exciting to listen to what's been done and what we dreamed about when we first heard about stem cells. So I was wondering about the bioethic limitations that are being currently discussed about using stem cells to create new organisms.
Yeah, it's a very good, very fair question. I have a good answer to avoid this discussion. My answer is that I'm technology enabler. I'm not telling you should do or shouldn't, okay? I know something very fundamental. If we don't act now on conserving, on cryopreservation of specimens, it will be the end of the story. It will never be possible. So I'm enabler. Then it's up to society. You, your peers, the government, the higher beings to decide whether they want to create more of these dolphins or not. I create the possibility. And I know that if I don't act now to create a possibility, that possibility will not exist. So that's what I know fundamentally. The ethical debate, we can discuss it a lot. I prefer to stay. I think it really depends on, there are so many animals, right? The animal, the biohabitat, and so on and so forth. So it's a very broad question, very hard to answer because there are different types of organisms.
But for example, to do the project that you were doing in the zoo, you need to have some permissions, like you said on your diagram.
Permits, yeah.
So I was wondering if there is already
this is highly regulated. I mean, a very regulated.
Exactly, the regulations behind it.
There's a lot of regulation. Actually, I think I'd say that the regulation has to change. So look, I mean, the regulation is in place in order to prevent trade of animals and animal substances like the rhino, parts, in many places in the world, they believe that they have these magic powers. So those things are in place in order to prevent animal trade and basically, sale over stocks, black market related things of animals. I think that you should actually differentiate what we do from that because because of all the regulation makes life very hard, very difficult. So there are Nagoya protocols and other protocols we need to follow. There's a lot of paperwork just to receive a sample from a whale. It's a lot of work. It's very expensive, and I don't think it serves the community of science at all.
Thank you.
Sure.
Thanks, Micha, for the fantastic talk. I don't know if you can comment a little bit about the positive association between longevity and body mass, the molecular mechanism.
Yeah, I mean, I intentionally didn't speak about this because I don't want to spoil things. And all those things are still in, basically in, we're working on this intensely. And this is also an open lecture, also online. I don't know how much competition is listening to us now. No, I'm joking a little bit. I think the question is really interesting, whether the intrinsic lifespan of cells is reflecting animal lifespan. And I don't have the answer yet, but I hope that if you invite me again in a year or two, I will have the answer. Okay.
Thanks.
Thank you.
Hi, thank you for the lecture. Given that reprogramming requires the erasure of original identity markers, yet epigenetic signatures can distinguish the induced pluripotent stem cells from their embryonic counterparts, to what extent do these retained epigenetic marks influence the efficacy or fidelity of the reprogramming process? Specifically, how might the persistence of such signatures from the donor cell lineage affect the differentiation from, well,
so basically the question, thank you for the question. The question is how much the tissue of origin has an impact on the outcomes of differentiation. I think this is the question.
Yeah.
Okay. We work on this a lot in the human context. We have large projects on that. We're spending a lot of money and actually mostly bioinformatics to look at that question. I mean, I don't know for the rhinos, I have no idea, and for other animals. But I can tell you this is, I mean, a very intensely sought after topic. I'll just give you an example of experiment one can do that we are doing. We took from one individual a few different tissues from one single person, okay, and made iPSCs from them, and then differentiate them as well. So comparing differentiated progeny to the epigenetic characteristics at the beginning. We took cells from the urine, the cells from the blood, cells from fibroblasts, and we are exactly following these questions to see if there's influence, trying to discern whether there's an influence coming from the tissue of origin. The answer is probably yes, but it also depends on reprogramming technology, passing many other technical details. So we're trying to control this very well by starting with one person, one reprogramming technology, and comparing genome from the get-go to the differentiated progeny, and we actually differentiating few cell types, and then comparing them across. So I don't know, but we will know. Again, invite me in 2 years, I will tell you. Yeah.
I would like to come back to the ethical question and to your escape, because at first I fully agree and I really subscribe to your declaration that us as scientists and technicians, we should not be the authority that decides about what should or should not be done, because this model would be equivalent to have a technocratic society, and this would something that I would hate. However, it's an easy answer to escape our own responsibilities, because we do have responsibilities in different ways, and I guess you're aware about it. One of them is that by creating possibilities, actually, we give the power to some society, and you might have benevolent societies and democratic societies, and you might have societies where the program is really antipathic and dangerous for human species and, or for society, for societal region. I mean, when you see that in China, the genome sequencing programs have been really fostered, but that an official declaration of the Chinese government was that they wanted to promote eugenic programs to select highly skilled and intelligent people, and so looking for the genes of intelligence and so on in the genomes, it's a bit frightening. And also an interesting example with George Church. You mentioned George Church, the capability to synthesize full genomes, and indeed he was very enthusiastic and communicating already in 2005, and this was the date where the first full chromosome was synthesized, and it was Mycoplasma genitalium. And he said at that time, within a few years, we should be able to regenerate full chromosomes of Neanderthal, and then even to make oocytes that, well, to make a zygote. And the only problem would be to find the carrying mother. And of course, this raised a huge debate because this is not the only problem. The problem is that by doing so, you will not regenerate an endangered species. You will create a human being who will be in a society, and not only in a society, who will be living with a species for which we have no idea what will be the skill of this individual. So I mean, there are huge questions. We cannot just escape them, and we need to be part of the debate. We should not be the deciders, but we should be the informers, including informing the society about the risks and the potential derivation of what we are inventing.
Thank you for this lecture. You're right. I mean, I'm completely on board. Yeah. But to me, I mean, very briefly, I have this perspective that if you want to create evil, you don't need biotechnology. It's very easy to create evil by few whatever pesos or whatever dollars, you can create a lot of evil. So biotechnology is not really the way forward. If you really want to become whatever, I mean, evil, yeah, you don't need that. So we know this very well in the world that we are living in now. So, but I agree with you that, I mean, we should be part of this debate. Yeah, totally. Yeah.
Okay, thank you very much. So you showed us that across mammal species, the bigger ones are the ones that live more. But I think that within a species, the smaller organisms are the ones that live longer. And I think that maybe that's because of some growth and proliferation pathways that can induce cancer in the bigger organisms. So in the sense of animal conservation, could you modulate these pathways in the IPCs so that you get mammals that live longer?
Okay, so you're asking basically, I mean, you don't have to, you're asking about longevity, if I understand correctly, the answer is that you're talking about the genetic tinkering or engineering in order to change traits. So basically, we all do this all the time. I mean, right? We have chickens that lay eggs every day and beef that is small or large. And so basically, we are all so natural selection, we do those things. I mean, people do. I mean, corn, in nature was this. Now it's like that, and so on and so forth. So everything we eat and culture in agriculture is completely engineered typically by natural breeding. If we can basically think, maybe make things longer or shorter, I think it's possible. Yeah. And probably it's possible. I'm not saying exactly how, but I would think it's possible. Yeah. Because there are species like naked mole rat, close to other rodents. It lives much longer than other rodents. Why? I think we'll figure it out. Yeah. And then you can use it. Yeah.
¿Alguna otra pregunta?
Thank you very much. Thank you very much. Well, regarding the ethical considerations, since they already addressed it, I'm going to address it as well. Wouldn't this kind of technology undermine any existing conservational efforts already in place? Because, well, if you can regenerate any species anytime you want, you wouldn't have to conserve it in the first place.
I would love this to be the case, but it's not. It's so hard. This is so difficult, right? That we are. I wish we were at the state where we can say, you know what, we can solve the problem. No problem. We are not. It's very far, and really, I think I mean, for most species, 99% of species, the way now is by habitat and so on and so forth. This is, I mean, in case of those other measures don't work. Okay, I'm not saying for replacement. And I think it's a mistake to say, okay, let's not discuss this alternative, because then you end up not having the dolphin or the rhinos and many others, and it will happen much faster than you think.
Right.
I mean, even for elephants and giraffes and so on and so forth, these large animals like lions and so on, how many of these iconic species we have? Specimens, more or less. Specimens of these iconic animals, all these African animals, there are maybe, I don't know, 1 million, okay? So it's basically, it's nothing. And they can go, many of the species can go like that.
Yeah, but those only in the supposition that these extinctions are human caused.
They are completely human caused.
Yeah, in those cases. But what about the species that are naturally selected? In those cases, should we apply this technology to save them from extinction and possible natural selection in this filtration process of evolution? or should we just let nature play itself out?
You know, we know more or less what's the general rate of extinction, okay? We are about at a rate that is, I mean, perhaps 1,000 times faster, okay? So I'd say that if you make, you know, simple math, you see that you are not in the range of what is expected ratio per year according to the history of history of history. So I think taking this in mind, I mean, it's really extinction on steroids now.
Right, thank you very much.
Sure.
Okay, thank you very much for your nice presentation, Micha, and for being here.
Thank you.